So I'm going to introduce Harris, Dr. Thea Herodies, who's our Institute for Neuro-Immune Medicine, professor of medicine, and has been very involved in working in neurodegenerative illness. He's going to chair this session, so I'm just going to hand it over to Harris.
Thank you very much, Nancy. Look, this is a real honor and pleasure to both have as well as have the chance to introduce Dr. Albensi, whom I've known in many capacities before I even came to NSU. I was at Taft in Boston for many years. In any event, he's a professor and chair of the Department for Pharmaceutical Sciences at the College of Pharmacy here in the NSU. He's also the editor-in-chief of the very esteemed journal Molecular Neurobiology, where I had also a chance and pleasure to publish a few papers. He got his PhD in neuroscience at the University of Utah Medical Center. Then he trained at Georgetown, and then at Sanders Brown Center of Aging, University of Kentucky. He'd been through a number of institutions like Cleveland Clinic, Case Western Reserve, and of course, he's associated with the University of Manitoba and the Hospital Boniface, as you can see on the slide. He's well known for his work in dementia, sort of Alzheimer's, and especially the role of NF Kappa beta in these diseases, and most recently in the potential role of mitochondria, with especially in Alzheimer's disease, with potential differences between the genders. So we're very excited to have you, Dr. Albensi. We're very interested in what you will say and looking forward to very successful collaborations pretty soon. So please go ahead.
That's great. Thank you for that kind introduction. Well, good morning or good afternoon, everyone, depending where you are. I'm in Alabama, so it's still morning here. I want to just start off by thanking Dr. Klimas and McKenna for inviting me to speak today. So I'm really honored to present at your Institute for Neuro-Immune Medicine seminar series. And today what I'm going to do is give you a broad overview of the research projects going on in my lab. And it's going to be a special focus really just on our investigations with mitochondrial function and dysfunction in Alzheimer's disease. And we have a lot of exciting new data that's coming out right now. So I'm really happy to share this with you. So these are my disclosures. So in the seminar today, I really want to talk about the increasing evidence for mitochondrial dysfunction playing a role in Alzheimer's disease and why my research really focuses on this target. And so to this end, I'm going to cover the following topics. First, we'll look at how central mitochondria are to really a plethora of diseases and talk then specifically about Alzheimer's disease. Then I'll show how brain metabolism and mitochondrial functions are disturbed in Alzheimer's. We'll look then at some of our data with sex differences in Alzheimer's disease that we've been publishing. And then next, we'll look at some of the results that we found with the inflammatory mediator that you may have heard of, nuclear factor Kappa B or NF Kappa B, and how some of our data suggests that it interacts with the complexes in the electron transport chain. And then we're going to show you our first attempt at some of our computer modeling, computational modeling of mitochondrial function. And then we're going to show some of the studies that we presented over the last few years, working with different diets, hormones, and drugs that modulate mitochondrial function. And then finally, I'll show you some of our new preliminary work with mitochondrial transfusion or transplantation. So what are some of the challenges that we face in the Alzheimer's disease world and community and with these patients? Well, importantly, it's important to know that throughout the world, someone's diagnosed with dementia once every 3 seconds. It's really a staggering number. Now, Alzheimer's disease is only one type of dementia, but it is the most common type. And as you can see in the table on the left, there are other types that include frontal temporal lobe dementia, Lewy body dementia, and vascular dementia. And of course, there are mixed forms of these too. Now, on the left, I created a table that shows different types of Alzheimer's disease, and this is not universally accepted, and it's not agreed upon by everyone by any means, but I mention this because I think the various types of subtypes of Alzheimer's, as I created in my list here, are something that many scientists aren't thinking about, and I think we should. So in my view, I think it's really critical for administering appropriate treatments as we go forward with our understanding. Now, in the United States, we have a lot of regional variation, and Alzheimer's disease is still on the rise, especially in some states. And so in Florida, where I am, of course, there's over 500,000 people that have Alzheimer's disease. It's really a staggering number. And what's even more staggering is that there's over 850,000 people that are taking on the role of caregiver. Now, in this figure, we see the rising rates for Alzheimer's depicted by the purple shading, and the very dark purple shading are the highest rates. So you can see this in states like Alaska, Nevada, Arizona, and Colorado. And it could be because these areas are more remote or rural, but we don't really know why the rates are rising in some of these areas. Now, Alzheimer's disease is also a leading cause of death, usually ranked around number 6. And you can see, as compared to other causes of death, we're really losing the battle since Alzheimer's is increasing. It's not decreasing, it's increasing, as shown in the bar on the far left. Whereas if you look at the percentages of other leading causes of death, they're either staying the same or they're decreasing. So this is a concern. But what causes Alzheimer's disease? And surprisingly, we still don't know after so many years of research. Typical symptoms include, of course, memory impairments, but there's also other cognitive and behavioral changes. And I saw this myself in my mother, who developed Alzheimer's disease. And there's also a lot of variation amongst people. Now, some of the pathological hallmarks of Alzheimer's you see in the illustration on the left, the sticky toxic amyloid plaques, and also the filamentous tangles that are associated with the protein we call tau, and especially when this protein gets hyperphosphorylated. But one of the major risk factors for Alzheimer's disease is actually aging. So it's important to keep that in mind, is that we're all at risk as we get older. And it's also a multifactorial condition. So probably a variety of things coming together. So like a perfect storm to create this condition. But it's also important to remember that in addition to the plaques and tangles, there are other important mechanisms and processes that still could cause Alzheimer's disease, and we just don't know for sure yet. But especially things like we study in my lab, such as inflammation and mitochondrial dysfunction. In fact, one of the hallmarks of aging is mitochondrial dysfunction, as you can see in this diagram showing the major contributing factors of aging. Now, given all the animal studies and some of the clinical studies that have been produced so far, many investigators have shown this sort of timeline. So this timeline shows the appearance of several different Alzheimer's disease hallmarks. And as you can see by the solid blue line, amyloid beta plaques seem to appear first, followed by changes in tau, which is the green line. And then eventually, this is followed by memory impairments that's represented by the orange line. Now, I'm hoping to convince you today why I think this timeline is incomplete, and we'll talk more about this later. So how do we treat Alzheimer's disease? Well, of course, before 2021, there were 4 main drugs to treat Alzheimer's disease, but none of these have been really that effective. And the top 3 were designed to improve cholinergic neurotransmission, and these drugs were also designed for the treatment of mild to moderate types of Alzheimer's. And the bottom drug, memantine, is actually targeting a different mechanism. This is trying to target the excitatory neurotransmission, the excitotoxicity that we see at the NMDA receptor. And this is approved for the treatment of moderate to severe Alzheimer's. Now, there have been some new drugs approved for Alzheimer's disease, but there have been some problems with these approvals. And all the new drugs are actually designed to block or clear amyloid pathology. So for example, Aduhelm was controversially approved in 2021, and it's now being discontinued by Biogen sometime in the fall this year. Also, lecanemab had accelerated approval in January 2023 and then full approval in July 2023, but there's no evidence that this drug actually improves memory. Then another drug that was expected to be approved last December in 2023 has been delayed, and this is donanemab, produced by Eli Lilly. Now, this drug still might be approved in 2024, but we just have to wait and see what's going on. So one has to wonder from some of this, is the amyloid hypothesis of Alzheimer's disease wrong? And I say this in part, especially when you consider the fact that one could have a head full of amyloid and not get Alzheimer's disease. So we're talking about a very complicated disease process. Now, of course, oxidative stress has been implicated in many age-related diseases, including Alzheimer's. But some of the antioxidant clinical trials so far have been largely unsuccessful as a preventative or curative measure. And also the clinical trials using anti-inflammatory drugs to treat psychiatric conditions, including Alzheimer's disease, have had limited success as well. So why is this? Well, we really don't know yet. And given some of these controversies with the new Alzheimer's drugs and the lack of overall effectiveness for the older Alzheimer's drugs, and also some of the limitations with antioxidant drugs and anti-inflammatory drugs, about 10 years ago, my lab took a different approach, and that's why we've been targeting and investigating treatments for Alzheimer's disease. So really, we started off this quest looking at brain metabolism. And so human studies have found decreases in brain metabolism as patients transition from a normal state to mild cognitive impairments, or MCI, and then to various stages of Alzheimer's disease, as you can see in these PET scans that look at glucose utilization. And what you can see that's really striking is the hypometabolic activity that's illustrated by the darker black areas that are shown in this figure, which increase in time as the disease progresses. However, little is actually known about the changes in mitochondrial activity in the living brains of patients with Alzheimer's. So there's really a lot we have to learn yet. And in this particular study, patients with early to moderate Alzheimer's were recruited and studied. And so in this study, they did find that mitochondrial dysfunction occurred in the parahippocampus, and this was in early stage AD. But we need to do more of this sort of work. Now, this is a human clinical trial that my team actually conducted recently, and we tested an intervention to see if it improved memory and other outcomes, and we used PET scanning here as well. Now, we haven't published this study yet, so I can't give you all the details. We're hoping to submit it sometime this summer. But we used PET scanning to compare those on the intervention that had MCI and compare those to that had MCI without the intervention. And of course, we used a non-parametric test to see if there's a difference between the two groups in terms of the percentage of metabolic changes. And you can see the colored shaded areas on the left that we did find some differences. So it was very interesting that in 4 regions, there were statistical differences between the two groups. And you can see that we graphed this with box plots in the figure on the right. And you can see in the group called QU, there was a decline in metabolic rate in these regions where the other TY groups showed an increase. So hopefully we can publish that this year and we can talk more about these interesting findings. Now, in the past, we've used PET scanning in our Alzheimer's mice as well. So to look at brain metabolism in the 3XTG model, we've used PET scanning actually with MRI. This was done by one of my postdocs, Ida Adlimigadam, who's now an assistant professor at Southern Illinois University. So here what we do is that we show a series of MRI horizontal slices in the mouse brain, and we overlaid the PET scanning images on top of the MRI scans. And the blue colored shaded areas that you can see in some of the top row really designate areas where metabolic activity has changed. So this is a nice approach that allows us to visualize both the functional changes in brain metabolism using PET, and then also to visualize the brain anatomy that's provided with the high-resolution brain anatomy that MRI provides. So in this experiment, fluorodeoxyglucose, or FDG, which of course is a radiopharmaceutical, was injected into both strains of mice, the control and the Alzheimer's strain. And you can see the colored shaded areas here show changes in FDG uptake. So here we use statistical parameter mapping, and we found that FDG uptake to be significantly reduced, actually, both in the insular and the piriform cortex in both hemispheres of the brain. Now, this is interesting to us because these regions are actually associated with olfaction and neurogenesis, and these might seem to be affected very early in the Alzheimer's disease process. And this data was published in the journal Neurobiology of Disease. So given that changes in brain metabolism in Alzheimer's have been found by several labs, including our own, and since mitochondria seem to be the major driver of brain metabolism, we've been spending a lot of our attention and time in my lab looking directly at mitochondrial function and dysfunction. Now, I'm sure everyone in this audience knows that the main functions of the electron transport chain of the mitochondria are to transfer electrons and create a biochemical gradient for protons or hydrogen. And these processes in the electron transport chain are coupled to the generation of ATP, as you can see here in the diagram. I just want to point out very quickly complexes 4 and 5 because they're of special interest. Complex 4, remember, is the site of oxygen consumption, and complex 5 is the site of ATP generation. And during metabolism, of course, you generate reactive oxygen species in the mitochondria, which is actually a normal byproduct of cellular respiration. But the idea that mitochondrial function plays a role in aging and disease is not actually new. And in fact, in 1956, Denham Harman proposed the free radical theory of aging, which he later modified in 1972 to be the so-called mitochondrial theory of aging. And then around 2004, Swerdlow and Kahn proposed the mitochondrial cascade hypothesis, which actually connected the mitochondrial theory of aging to Alzheimer's disease pathology. And this hypothesis was very interesting to me, and this is how I got into this about 10 years ago, because it really emphasized the importance of mitochondrial dysfunction in the pathogenesis of sporadic AD. And so Swerdlow argues that amyloid might be triggering some of the changes in mitochondrial function, but we don't know for sure, and it could be the other way around. So that is, mitochondrial dysfunction could be also triggering amyloid in some ways. But in any case, mitochondrial dysfunction seems to be linked also to NF Kappa B, or nuclear factor Kappa B activation, which is a major mediator of inflammation. And we're going to talk more about that later in the seminar today. And Swerdlow actually acknowledged this in his theory in this paper that was published back in 2018. But in spite of this history, what I want to emphasize is that the role of mitochondria and mitochondrial dysfunction, I think, has been underappreciated for many years. So, and it's curious to me because in fact, when you look at diagrams like this, I was a board director for Mito Canada for 6 years. It really kind of just jumps out at you how important mitochondria are. So you can see that it's central to many disorders and diseases, over 30 actually, and it doesn't even include some of the the affective disorders. But we can divide these sorts of conditions into the primary mitochondrial disorders and also the so-called acquired mitochondrial dysfunction. And the primary mitochondrial disorders are usually genetically inherited, and those are diagnosed by identifying mutations in mitochondrial DNA, and they're represented here by the dark green circles. So with the acquired mitochondrial types of dysfunction, this is more common to us, and we can see those shown in this diagram with the light green circles. And these are conditions that could be genetically influenced or by also caused by environmental factors, or really a combination of both, which is more likely. But the acquired dysfunction can include more common conditions like diabetes or cancer, Parkinson's, and even Alzheimer's disease. Now, mitochondria, of course, have many normal functions besides just ATP production, and we don't have enough time to go into all of those functions now, but there is one other function that you should know about, and that is that mitochondria are mobile. This is a more recent, really, observation. And my former research associate, Dr. Sabir, who's now an assistant professor at Nova Southeastern University, explored some of the mitochondrial biodynamic mechanisms as shown in this video that he created. So mitochondrial mobility is really important for a lot of reasons, and changes in mobility can really have important implications, not just for ATP output, but also for memory and memory encoding. And you have to remember that synaptic plasticity, of course, requires energy, and it's a high energy demanding process. So this video shows the motility of mitochondria, which occurs along microtubules. The mitochondria are moving to sites of high energy demand, such as synapses. They're actively encoding new memories. So any disruptions in some of the proteins and processes that the mitochondria make that regulate mobility can really affect memory encoding. Now, over the last 10 years or more in my lab, we've observed a number of different types of mitochondrial functions affected in Alzheimer's disease. And I kind of alluded to some of this already. This includes decreases in glucose metabolism, increases in reactive oxygen species above normal levels, changes in axonal transport of mitochondria along the microtubules, changes in the shape and size of mitochondria, also alterations in quality control or mitophagy, a disruption in the biodynamics, such as in these fusion and fission proteins, decreases in mitochondrial numbers, which has a direct relationship to ATP output, and also deficits in these complexes of the electron transport chain that we'll look at more closely in just a few minutes. Some of our work has also looked at sex differences in Alzheimer's disease, and this is something that we really kind of just accidentally jumped into a few years ago. It's important to note there are sex-based differences in the occurrence of Alzheimer's disease, and we can't yet fully explain this. And in fact, about 2/3 of those with Alzheimer's in the US and Canada are actually women. So why is this? Well, it is known that women live longer than men, or we used to believe that statistic. We don't know if that's going to continue to happen going forward. There are differences in brain structures, of course, and there might even be differences in psychosocial stress responses. Of course, there are sex hormone differences, and there could be important differences in the inflammatory response and in the immune system, and then, of course, genetics. Now, interestingly, all of your mitochondrial DNA actually comes from your mother. And so it's through a maternal lineage. And this could also have some influence on sex differences in Alzheimer's disease. Another reason for sex differences might have to do with sex hormones. And sex hormones in general decrease as we age, as you can see by the solid red and blue lines in this diagram. But the amount of decrease varies tremendously depending on what sex we are. And you can clearly see that male hormones such as testosterone, or the blue line, taper off gradually as men age. But then in females, there's an abrupt decline in estrogen, the red line, following menopause. And in fact, before menopause, women are actually at lower risk for Alzheimer's than men. But after menopause, their risk triples. Now, in this graph, we also see some dotted lines. These are lines showing redox levels, and these correlate to changes in sex hormones. So what's happening overall is that these redox levels begin to rise as we age. And you can see that the rise is different depending on whether you're a male or female. So hormonal deficits in women and men are another risk factor for Alzheimer's disease. And because of this, you might ask about the benefits of hormone replacement therapy, or HRT. And as far as I can tell from the physicians and scientists that I've met and discussed this with, really this research has shown mixed results. And I hear a lot of different opinions yet on HRT. But really the mechanisms that are underlying sex-dependent neuronal changes are still not well understood. So to this end, our lab started looking at some of this about 7 or 8 years ago, and my postdoctoral fellow, Dr. Jelena Dvorževic, shown here, led some of these initial experiments investigating potential sex differences. And in some of our early work, we used 2 different strains. We used the 3XTG Alzheimer's transgenic strain, and we also used the TG-CRND8 strain, another widely used transgenic model of Alzheimer's. Now, in studies with our 3XTG mice, she found changes in several mitochondrial proteins and their expression levels. So, for example, on this slide, she showed both age-dependent and sex-dependent changes in a protein called dynamin-related protein 1, or DRP1, which is a mitochondrial protein known to regulate processes of fission. Moreover, when we examined mitofusion proteins, the regulators of fusion, we again found age-dependent and sex-dependent changes in the expression of these proteins in different regions. So overall, when examining DRP1, MFN1, and 2, we found a shift towards more fission. Now, fusion and fission occur when cells experience metabolic or environmental stresses. Now, fusion is interesting because it helps mitigate stress by mixing the contents of partially damaged mitochondria as a form of complementation, where fission, on the other hand, is needed to create new mitochondria, but also contributes to quality control. And it seems to enable the removal of damaged mitochondria and can facilitate apoptosis during high levels of cellular stress. So disruptions in these proteins and these processes can affect development and have been implicated in a variety of neurodegenerative diseases. We also looked at one of the uncoupling proteins, UCP4, and they have several functions. And in this study, we also found changes in expression levels that were age-dependent and sex-dependent. And disruptions in these uncoupling proteins can affect mitochondrial membrane potential and also ROS levels. So the other thing that we did is that we measured oxygen consumption rates, or OCR, and we did this in either cell cultures or tissue homogenates. And we analyzed these samples, in this case, using this instrument called the Seahorse XF24. Now, the Seahorse XF24 is a high-throughput plate reader system. And I'm excited that we're getting a new one here at NOVA sometime at the end of the summer. Now, you can actually measure two mitochondrial parameters. You can measure OCR or ECAR. And now ECAR stands for extracellular acidification rate, which is actually an indirect measurement of glycolysis. Now, this figure shows our standard operating procedure, or SOP, when using cells, and we call it the mitochondrial stress test. And what it does is that it allows us to determine several parameters of cellular respiration. So in this version of the test, we administer 4 different drugs that you can see listed on the top that modulate different aspects of mitochondrial function. So for example, when we add ogleomycin, this has an effect at complex 5 at the site of ATP synthesis, and this reveals coupling efficiency. When we add FCCP, it actually creates pores in the membrane of this mitochondria, and this uncouples respiration. So it uncouples the respiratory chain from complex 5 and reveals maximal respiration. And then finally, we add 2 other agents. We add rotenone and antimyosin A, and rotenone is known to inhibit electrons in complex 1, and antimyosin A will block electron transfer at complex 3. So no further oxygen can be consumed at complex 4. So that's what we do in cells, but in our mitochondrial experiments using tissue homogenates, we use a slightly modified version of this, and we add one other drug. And what we do is that we add ADP to energize this system before we add the other compounds I just showed you. So anyway, so in these experiments, we used these sorts of procedures, and we isolated both cortical and hippocampal mitochondrial samples from males and females. And in this case, these were adult TGCRND8 mice. And what we found is that we found increased basal and coupled respiration in the female hippocampal samples, but not in males. And you can see that indicated by the green circles. We also found decreased uncoupled respiration in both female and male cortical samples, and I show that here with the red circles. Now here I show a different study. This is in the 3XTG mice, and we again compared males to females. We also sampled 2 regions, the hippocampus and the cortex, but in this case, we did it in 3 age groups. So we did it at 2 months of age, 6 months of age, and 14 months of age. This study took a couple years at least to obtain all the data. And these graphs that you're looking at are just a representation, actually, of many experiments. And the red arrows, the red block arrows, are indicating sets of data that had significant changes, okay? So what we found is that we found decreased mitochondrial function very early in the cortex and hippocampus, as you can see on the right side, in the female mice at 2 months of age. However, in the males, we didn't see any OCR deficits appearing at all at 2 months of age. In fact, we didn't find any deficits until 6 months of age. So overall, the cortex seemed to be more susceptible to mitochondrial changes than the hippocampus. And you can see clearly that there's a different pattern of OCR deficits between males and females. And this paper was published in the journal called Cells. We also used a second instrument to verify some of these results, and this is the Oxidative 2K. And this allows you also to measure OCR. Now, this is a port injection system. It's a little bit more labor intensive, but we can actually measure 5 different parameters overall. So in addition to OCR, you can measure ATP, mitochondrial membrane potential, ROS levels, and even mitochondrial calcium. But we used it to measure OCR and hydrogen peroxide, a type of ROS, and also to look at ATP changes. Now, it's interesting because you can also use this instrument to measure mitochondria and white blood cells. And so you can take human blood samples, making this instrument useful for human clinical trials. So using this instrument, we found that OCR was reduced in female hippocampal samples from these TGCRND8 mice, but not in males of the same age. We also looked at mitochondrial complex subunit proteins that you can see here, and we found deficits in complex 1 and complex 4. However, when you compare the results between males and females, we found that only the females were showing deficits in the expression of these subunits. Now, in the other model that I talked about, the TGCRND8 mouse model, we also found that the inflammatory factor I mentioned earlier, nuclear factor Kappa B, had subunits that were also expressed differently in males versus females, as you can see in panels A and B. And we looked at 3 different subunits, and we saw differences in P105 and P65. And I'll come back to some of this in a few minutes. But what causes these mitochondrial deficits? Well, one possibility is amyloid beta directly or indirectly can activate NF Kappa B. So that's one route, and this could cause the released dimer, the active NF Kappa B, to go into the nucleus, and then to turn on or turn genes off, and triggering a variety of different pathways, which can then indirectly affect mitochondrial function. The other model is that mitochondrial deficits could be caused by amyloid beta entering the mitochondria at these TOM sites. These are pores, and the TOM stands for translocase of the outer membrane. Now, once inside the mitochondria, A beta might actually interact directly with specific sites in the mitochondria or molecules in the mitochondria. And some studies show interactions of A beta with molecules such as ABAD, or amyloid beta binding alcohol dehydrogenase, which has been shown to increase levels of ROS. Other sites of interaction have shown to be cyclin D and other sites on the electron transport chain, just to name a few. So to this end, we've begun to measure various forms of amyloid in both cytoplasmic and mitochondrial compartments. We still have a lot more to do, but so far, we found sex-based differences in the appearance of amyloid oligomers when testing mice that are 6 months old, and we have a lot more to do with different age groups and different strains. But overall, these data show increases in amyloid in females, but not in males at 6 months of age. We have also been testing applications of A beta in cell cultures, and here we find that various doses of amyloid beta increase NF Kappa B subunits such as P65 and I Kappa B. Also, in the slide on the right, or in the diagram on the right, we found decreases in cell viability when A beta was applied for various durations of time, up to 3 days of exposure. Okay, so let's look a little bit more closely now at factors like NF Kappa B that we found affect complex 4 in Alzheimer's. So we mentioned a few times in the slides we showed earlier, but you can see that there, like I said, there are 3 subunits, the I Kappa B and typically the P65 and P dimer. So when I Kappa B is degraded by an upstream molecule, this dimer, very typically the P65 and P50 is released, and this translocates to the nucleus where it can turn on different genes or it can turn off certain genes. So from a biochemical signaling point of view, NF Kappa B is really a key regulator of inflammatory responses. And you can see some of the potential activators of NF Kappa B here. It includes infectious agents and cytokines, and also factors that stimulate the immune system also activate NF Kappa B. So there seems to be a variety of responses that are possible once NF Kappa B becomes activated, and this includes the production of even more cytokines and inflammatory responses, as you would expect, and even the conversion of short-term to long-term memory, but that's really the subject of another seminar. So this has tremendous impact on turning on different genes in various networks. Now, we can see here that some of the additional steps that are involved in the inflammatory response once NF Kappa B is activated. So during step 1 or priming, you can see that the role of NF Kappa B and NF Kappa B activation leads to the transcription of other molecules such as NLRP3 and interleukins. And following this is yet another step involving the inflammasome, which there's a lot of literature growing that I find on PubMed these days talking about inflammasome assembly. But inflammasomes are part of the inflammatory response and appear to promote proteolytic cleavage and secretion of cytokines such as interleukin-1 beta and interleukin-18. Now, importantly, NF Kappa B has also been found to be co-localized with amyloid plaques in human brain, and these have been found in Alzheimer's patients. An amyloid precursor protein, or APP, which is the parent molecule of A beta, or the A beta fragment, is also a gene target of NF Kappa B. Also, NF Kappa B subunits have been found in the mitochondria by Cogswell et al. Actually back in 2003. So we've known about this for a while. So using Western blot techniques, my lab, we show here decreases in the expression of some of these subunits, in this case, NDU FB8 in complex 1 shown on the left, and also in the subunit MTCO1 in complex 4 in our 3XTG mice. So in other words, you can see decreases represented by the blue bars in the 3XTG mice as compared to the purple bars that represent the control mice. So let's take a little bit of a closer look at complex 4. And you may or may not know that COX1, COX2, and COX3, circled in blue here, are actually encoded by mitochondrial DNA. Now, some studies suggest also that A beta interacts directly with this complex, and it interacts with complex 1. Now, a study by Shi a few years ago implicated NF Kappa B in complex 4. And you can see in the left figure, panel A, the top left corner, there's an MTT cell viability assay, and this was used in HT22 cells. And what they did here is they added a variety of doses of A beta that induced mitochondrial dysfunction. And you can see that increases in doses of A beta actually lower the MTT viability index on the y-axis. However, when this NF Kappa B blocker, Bay11 7082, was applied, Bay11 was seen to block the A beta induced dysfunction. And this is shown on panel A. And it also improved ATP levels as seen below it in panel B on the left. Moreover, when Bay11 was applied on the right side, in the figure on the right, it also was found to reduce COX3 activity. So I just showed you COX3 a few seconds ago, and that's a specific subunit that makes up complex 4. So altogether, overall, applications of Bay11 7082, when applied, reversed the A beta induced reductions in COX3. Okay. Now, using a different NF Kappa B inhibitor, SN50, which is a more downstream inhibitor, we also showed decreases in OCR. So this implies that there's some sort of influence on complex 4 function. So taken together, these data suggest an interaction between NF Kappa B and complex 4 that really needs further study. And for further discussion on this, you might want to read a review paper I published a couple of years ago looking at NF Kappa B activity in the mitochondria. But because of this activity, because complex 4 subunits were affected in both animals and humans in Alzheimer's, because of the interactions that we've seen in NF Kappa B on complex 4, we've decided to study complex 4 further biologically. And we're also in the process of attempting and developing a computer model of complex 4 that my postdoctoral fellow, Dr. Eni, presented back in March to your institute. So it's interesting because models have already been created to model oxidative stress in the mitochondria. So I'm not going to go into great detail about our model since Dr. Eni just presented this, but the history of this work is as follows. So my former biomedical engineering student, Chris Kodanik on the left, really started the development of this model, and this became his MS thesis project. And then Dr. Eni, my current postdoc in Canada, is now picking up where Chris left off. So some of the validation steps for this model by Chris and Dr. Eni includes measuring oxygen concentration in biological samples. We did this in mouse brain. And this is in response to the mitochondrial stress test that I explained to you earlier. In this case, we used the Ouroboros Oxygraph 2K to get our oxygen concentration data. And you can see here in this slide that oxygen concentrations that are represented by the dotted blue line can be seen here to be decreasing over time. So it behaves differently than OCR, which is represented by the red line. And you can see is going up and down and responding to the administration of these drugs that we use in the mitochondrial stress test. So what we want to do, of course, is to try to compare the experimental data to the computer modeling data. And that's what Dr. Eni has been doing lately and shown in this figure here. So the red line is actually shown to be very modeled very well as compared to the black line, which represents the computer-generated oxygen levels. So why are we doing this? Well, we're hoping that we have some sort of predictive value over some of these parameters like OCR. So another thing that Chris and Dr. Eni both have worked on is a GUI, or a graphical user interface, that allows the user to supply input for various functions. So one application of this model might be able to predict factors such as OCR under various conditions. And we're still working on this, and we're hoping to publish this paper sometime later in the summer. So what I want to do now is talk a little bit more about some of the other data coming out of the lab the last few years, where we've tested a variety of diets, also drugs, and also hormones to modulate mitochondrial function. Now, currently, there aren't any FDA-approved medications for treating mitochondrial disorders. However, there are a host of experimental therapeutics and treatments being tested for both the primary disorders and also acquired mitochondrial dysfunction. The figure shows some of the compounds that have been used in the past to treat the primary mitochondrial disorders and so-called mitochondrial cocktails. But you should also know that there are several companies around the world working on new mitochondrial therapeutics that will have application probably to both primary disorders and acquired mitochondrial dysfunction. So stay tuned for that. But our work on the modulation of mitochondrial function so far have included creatine, nilotinib, and estradiol, and that's what I'm going to talk about now. So in this study, my postdoctoral fellow, Dr. Wanda Snow, tested mice fed a creatine diet for about 8 weeks. And creatine, of course, I'm sure you've heard of it, is an over-the-counter product that you can get at the drugstore. But we also consume creatine when we eat meat and fish. And bodybuilders, of course, have been using creatine for many years to rapidly build muscle mass. So in this study, she found that creatine improved learning and memory, as tested in the Morris water maze, in normal mice. And she also found that it actually improved OCR when we measured it in the seahorse and control mice. But however, when she used an Alzheimer's strain, the 3XTG model, we also found that creatine had benefit, but again, the benefit was sex-specific. So the creatine supplementation showed differences on several parameters on spatial cognition in the Morris water maze. And she also found improvements in mitochondrial function that were sex-specific with regard to different parameters of mitochondrial respiration, as we can see here in the seahorse. And you can see that in the bottom row, you can see there's an increase in the gray bar. So every parameter measured in the seahorse actually was improved by the administration of creatine. Also, other protein biomarkers were assessed. We looked at markers that play roles in plasticity and memory, also in Alzheimer's disease pathophysiology, and also we looked at some inflammatory markers as seen here. Now, some of these markers included I-Kappa B, PCREB, calcium calmodulin kinase 2, and a synaptic marker PSD95. So overall, the data from this study showed that creatine really increased many of the plasticity-related proteins, such as calcium calmodulin kinase 2, PCREB, and PSD95. And it also increased the NF Kappa B suppressor subunit, the I-Kappa B. So when this subunit is increased, it usually shows that there's less NF Kappa B activation. She also found that it decreased different amyloid beta trimers and also tau, these pathological hallmarks of Alzheimer's. Now, it's interesting because most of the effects that we found actually showed the beneficial effects were shown in females and not males. And you can learn more about this study by going to our publication in the journal called Nutrients. Now, this is another study. My postdoc, Dr. Ida Adlimbagadam, she worked for me in Canada for about 6 years. She's a new assistant professor at Southern Illinois University in Springfield, Illinois. So she led this study. And so she used the anti-cancer drug, the repurposed drug called nilotinib. And nilotinib is a tyrosine kinase inhibitor. So it's a medication that's actually used to treat leukemia. But we got interested in this drug because our colleagues at Georgetown found that it actually had benefit in treating Lewy body dementia and Parkinson's dementia in a clinical trial at Georgetown. This was done and led by my colleague, Dr. Scott Taylor at Georgetown. So related human work with nilotinib is really being conducted right now, and they have a variety of different tyrosine kinase inhibitors that they're testing. But anyway, so Scott encouraged me to test this in our animal models, and this is what we found. So to look at the effects of nilotinib in the 3X mice, we looked at OCR, and Ida tested both neurons and astroglia in these samples from 3XTG mice. And we found that 100 nanomolar nilotinib was actually optimal for improving several mitochondrial parameters in the seahorse. We also found that the number of mitochondria increased in 3X mice in their astroglia when we used electron microscopy using the same dose, 100 nanomolar. And this is interesting that it also increased mitochondrial numbers. We also found it increased the expression levels for some of the biodynamic proteins, such as MFN1, but not DRP1 that we mentioned before in one of our other studies. And then we also found that nilotinib actually reversed the complex 4 subunit expression deficits. Lastly, we looked at the effects of nilotinib, and we found that they were somehow linked to NF Kappa B. And to our surprise, this inhibitor I mentioned to you before, the BA11-7082, we showed that this inhibitor reversed the enhancements of 100 nanomolar treatment. So in other words, the nilotinib-induced OCR increases were reduced in the presence of this NF Kappa B inhibitor, BA11-7082, and we can't fully explain that yet. Ida also did some Western blotting experiments, and she showed that there were increased levels in calcium calmodulin kinase 2, PGC1 alpha, and NRF2 in the 3XTG astroglia with different, she tried a variety of different treatments. This is with 20 hours of treatment, 24 hours of treatment at 100 nanomolar. So overall, these data suggest that nilotinib increases the number of mitochondria, and it seems to involve this calcium calmodulin PGC1 alpha NRF2 pathway that we sketch out here. So taken together, this study really supports a role for nilotinib in improving mitochondrial function overall, and perhaps astroglia could be thought of as a key therapeutic target in treating Alzheimer's. And this study was published in the journal Aging and Disease. Now, what I want to do now is to show you some of the new data coming out of my lab in Canada. I still have some people working up there, and this is by my PhD student, Pranav Mishra. And he's also investigating sex differences. Now, unfortunately, his work was interrupted by COVID, and we were shut down for a really long time. And so he's only had time so far to look at female hormones such as estrogen. Now, the estrogen hormone family contains 3 different forms, but he's really been focusing his attention on 17-beta estradiol. Estrogen receptors are actually found in both women and men, and they're distributed widely throughout the brain, as you can see in this diagram. And there's 2 types. There's estrogen receptor alpha and estrogen receptor beta. And these receptors seem to be predominantly found in the cerebellum, the ventral tegmental area, the hippocampus, the amygdala, and the frontal cortex, as well as in the raphe nucleus in the midbrain. And there also seems to be a greater concentration of estrogen alpha receptors in regions such as the amygdala and the hypothalamus, where estrogen beta receptors dominate in the hippocampus. So the estrogen receptor alpha and estrogen receptor beta, I should also mention, have been found in the mitochondria, okay? Now, in one of the recent experiments that he conducted in neurons, Pravda showed that estradiol actually increased OCR in a time-dependent fashion. However, he didn't find any change in ATP levels. He also showed that estradiol had a positive effect on AMP kinase, which is a cellular energy sensor studied extensively in diabetes. And moreover, he found that estrogen increased PGC1 alpha, the molecule we talked about before in Ida's work. And of course, this is a molecule that's involved in mitochondrial biogenesis. And he also found that the NF Kappa B subunit, I Kappa B, the repressor subunit, was also affected by the application of estradiol. The other thing I want to talk about is that people talk about type 3 diabetes, and it's a term used loosely, and it's a bit controversial, but some researchers use it to describe the theory or idea that insulin resistance and insulin-like growth factor dysfunction in the brain actually might cause Alzheimer's. So, but you know, using this term is a bit controversial. But while in Canada, I had the opportunity to collaborate with a diabetes expert, Dr. Paul Fernyhough. And one of his models for diabetes included the dorsal root ganglion, or DRC, cell culture model that you could see here, shown on the right in the slide. So these dorsal root ganglion neurons are used as an in vitro model for diabetic neuropathy. And of course, diabetic neuropathy is a complication that we see often in diabetes. But these DRG neurons are linked to are very sensitive to metabolic imbalances that occur in diabetes. So this serves as a very useful model. So Pranov used this model, and in this DRG preparation, he first determined that the E2 receptors alpha and beta are both localized in DRG neurons. He then applied the ER alpha agonist PPT and found that it improved OCR in the DRG preparation in a dose-dependent fashion. He also tested PPT and the other agonist, ER beta agonist, DPN, but only PPT increased AMP kinase as shown on the left, and not with DPN as shown on the right. So we conclude then that estradiol acts first via estrogen receptor alpha to activate AMP kinase and improve mitochondrial function in these adult DRG neurons. And we have some other data that's coming out that supports this too. We just haven't published this yet, so I can't show all the data right now. So this is the pathway that we suspect is being activated with applications of estradiol. And you can see in some ways it is a bit similar to what Ida had been finding and thinking with the administration of nilotinib. Now, another therapeutic approach that we've tried in the lab involves mitochondrial transfusion or transplantation. And so for a short history of mitochondrial therapeutics and mitochondrial transfusion, see my new editorial that I just wrote a few months ago that goes through some of this history. But in this slide here, I just want to describe very quickly the proposed idea on so-called mitochondrial transfusion. And this slide was provided courtesy of my collaborator, Tom Benson, who's the CEO of a company called Matrix Bio. Now, in this approach, one way of doing this is to extract stem cells and isolate healthy mitochondria. And then the idea here is to take the mitochondria and grow them in a bioreactor, such as what Tom uses, creating larger quantities in a pretty rapid fashion. Then the idea is to take these healthy mitochondria and transfuse them into animals or patients, which would increase very rapidly mitochondrial numbers. And so by increasing the number of healthy mitochondria, one should be able to both weaken the effect of the damaged mitochondria and also increase ATP output. Now, my lab hasn't used the bioreactor approach yet, but what we have done is shown here. And this is a study we published not too long ago. So this study was also done by Ida. And what we show here is these are, using Western blotting techniques. We compare the expression of protein subunits at 1 month as compared to protein subunits in 12-month-old animals when we looked at different subunits in the complexes of the electron transport chain. We sampled both cortical and hippocampal regions. And overall, what Ida found in this is that aging reduced the expression of key protein complexes in complexes 1 through 5, as you can see in all these histograms. So in other words, when comparing the black bars of the young mice to the gray bars of the old mice, you can see that the protein expression of each of these subunits go down in every case. However, when we took mitochondria from the liver from 1-month-old animals and injected these mitochondria IV into older mice at 12 months of age, we found an enhancement in one of the mitochondrial complexes, that is complex 2, that can be seen here in the diagram in the bottom right panel. And that compares not only a couple doses, but it compares it to the control levels. So this was found to be beneficial at both of the doses that we tried. And of course, this data is just a start, and we need to do more work, not only in aging mice, normal mice, but also in larger animals and in more models of Alzheimer's disease. And we're hoping to do that work in the future, sooner than later. So let's summarize. So today I discussed the fact that some of the older Alzheimer's disease drugs only show modest benefit, and some of the newer drugs have tried to clear amyloid, but some of these drugs are problematic in some ways. We also talked a little bit about antioxidant and anti-inflammatory drugs not having done as well as people had hoped. And I also provided a rationale and evidence for why I think targeting brain metabolism and mitochondria might be a better approach to treating Alzheimer's disease. So from the data I presented today and other evidence, we have to ask this question: is this the cycle we should target? That is, interactions between mitochondrial function and inflammation. So if that's the case, is it the inflammation that's affecting the mitochondria in a negative way? Or is it mitochondrial dysfunction that leads to increased inflammatory responses? And I think we really don't know yet. But let us assume just for a moment, for the sake of argument, that pathology flows in this direction. So that is that mitochondrial dysfunction leads to increased inflammatory responses. So what mitochondrial mechanisms might we target? Well, there are a lot of them. And many of the mitochondrial products that are released from mitochondrial dysfunction seem to drive inflammatory responses. And some of these are referred to as DAMP, and DAMP stands for damage associated molecular patterns. But many of the mechanistic links have not been determined yet. And so there's a lot of work to be done establishing links between mitochondria and inflammatory responses. But in the studies today, what I showed you evidence for creatine, for nilotinib, and estrogen for improving mitochondrial function. And I also showed you a novel method of using transfusion as a way to quickly increase mitochondrial numbers, potentially improve mitochondrial health. Now, some of the data also showed that improvements in mitochondrial function also reduced inflammatory factors such as NF Kappa B. I mean, we need to do more work, of course, at looking at other inflammatory factors, such as the inflammasome and some of the interleukins and other various species of ROS, and that is yet to be done. But our data showed improvements not only in markers of plasticity and tests of memory. So this suggests that by improving mitochondrial function, we can also reduce inflammation and improve memory and cognition. So it stands to reason that these results might be generalized to other diseases too, such as what you folks study in your institute, such as Gulf-Fuller syndrome, and also other conditions such as brain injury, Parkinson's disease, ALS, and even astronaut health, since spaceflight seems to accelerate aging. So I hope I've convinced you today that mitochondrial dysfunction in Alzheimer's disease has really been underappreciated, and it should be considered as a key target for treating Alzheimer's disease. I also showed you evidence that there are sex-based differences that exist in mitochondrial dysfunction in our different Alzheimer's mouse models. And mitochondrial dysfunction begins very early overall, and even earlier in females. So given this data, given the animal data, given the human data, what it suggests to me is that we really need to modify this timeline, and we need to add mitochondrial deficits as a very early event in the overall scheme of disease progression, perhaps before amyloid plaques. So the data also suggests that mitochondrial medicines and therapies for Alzheimer's need to be sex-based or personalized. and that the electron transport chain complexes have been affected by a variety of factors, especially complex 4. And I showed you evidence that dietary factors, drugs, hormones, and transfusion can improve brain metabolism and a variety of mitochondrial functions. And then finally, some of the pathways that we've identified or implicated so far are important signaling pathways that include PGC1 alpha, AMP kinase, calcium calmodulin kinase 2, and NRF2, in addition to the inflammatory pathway NF Kappa B that could be targets for improving mitochondrial function. So our future work in the lab will look more closely at some of these pathways that have been implicated and illustrated in today's seminar. So I think I'll stop there. I want to thank all my funding sources over the years, especially more recently, that supported the work I just showed you, especially recent funding that we've acquired from the NIH and also from the Alzheimer's Association in the US that Ida is using, that allowed her to transition to Springfield. And so thank you for listening. And with that, we'll stop and we'll take questions.